And if you can start, that would be great, and I can see your screen perfectly.
Okay. Thank you, Alison. Hello, everybody. I'm Arda, co-founder and CTO of Nanomik Biotechnology. As Nanomik Biotechnology, we are producing next-generation micro-encapsulated biopesticides for the agriculture, sustainable agriculture. So, before the Nanomik, I'm also a bio-entrepreneur, and I have a background of the molecular biology, and also I'm doing my PhD about the developing of the protein-based biopesticides. And Nanomik also founded in 2015 by co-founders, me and my co-founder, and up to now we closed 60K pre-seed investment, and we have investors from Denmark, Netherlands, and Turkey. We are developing a baseline technology, micro-encapsulation baseline technology. But in the meantime, we are also developing new biomolecule discovery systems, and we have products for pre-harvest usage and post-harvest usage. And up to now, we also collaborated and awarded from different kind of companies, and also we are part of the EIT Food and EIT Climate, and also we are we have prize from the Biomimicry Institute from United States. So before the technology, what is the idea to start this business for us? The main idea is that we inspired from the plants, because plants have able to produce volatile molecules, and these molecules can act as a everybody knows that as a activity against fungi, insects, or herbivores. But in the meantime, these molecules can able to have specialty to warn other plants in the neighbor to protect themselves against these pathogens in time by time. So of course, in the industry, in the product development industry, there are lots of products which comes from this specific herbs and plants and used as a botanicals against pathogens. But the general problem of this type of biopesticides are their stability. They have a stability problem, persistent problem, and they have to be used multiple spraying times. So this caused low price performance, and they don't show stable activity season by season. So this is the general problem of this type of botanical molecules. So because actually why they couldn't able to show standard activity in season by season, because they directly affected from the physical and chemical factors in the environment. So these volatile molecules can be sensitive against UV degradation, oxidation, and also heat degradation, and they lost the activity. Then they couldn't have enough time to act as a biopesticide in the on the plants. So beyond these problems, we as a company, we want to understand their combinatorial synergistic activity against specific pathogens, plant pathogens, and we started to understand this whole biomolecule, plant-based volatile biomolecule library, and we tested their combinatorial effect in our high throughput screening systems. So, and we found lots of specific combinations in the whole plant library, plant molecule, volatile molecule library, and then we tested again and again. But we understand that these molecules have to be protected when they sprayed to the crops. So then we developed our microencapsulation microsome technology. So what are we doing? We are the specific volatile molecules we are putting in a small microcapsules, and we are gaining them as a controlled release mechanisms in different kind of chemical and physical trigger conditions. So this technology also patented, and we have Deep Tech Award from Climate Launchpad. So the advantage of our technology is it's not specific for the plant secondary metabolites or volatile metabolite. It can also be used for the antimicrobial peptides or proteins or specific capsule proteins. So these capsules, when they apply to the crops, these capsules control the release in different pH conditions or the water volume. So, and it gives longer shelf life to the molecules, and also it's very suitable for the all different pesticidal application, fungicidal or insecticidal. And when we compare with the traditional encapsulation technology, our technology allows a high rate encapsulation efficacy, and the machinery also easy to scale up. So the capsules which we generated, the diameters almost up to 5 micrometers, and the frequency is so narrow, and it's the specific size of these capsules directly affects on the pesticidal activity, because the similar size of the capsules can able to penetrate plant tissue in the same time. So in our studies, we found that in different pH conditions, we tested in different pH conditions. In acidic pH, our capsules release more active ingredients than compared to the neutral pH. So these capsules it gains the tunability effect. So when you use the pH conditioner in the spraying tank, you can it depends to the conditions, disease rate in the field. You can use as a preventive, also curative product. And also in neutral pH, capsules are more closer to the zero order kinetics. So the water content is also important. When the water content in the microenvironment of the tissue of plant is high, the capsules going to be swell. It's getting bigger, and the porosity on the surface of the capsules it's increased, and the active molecules can able to diffuse outside. So our capsules directly affect on the loss of cell wall integrity. So when the capsules attach to the surface of the fungi, so it's start to control release, and the biomolecules which already we used have an effect on the membrane, and it start to disrupt and also start the ion leakage from inside to the outside of the cells. So when we compare with the traditional botanical formulations, actually they have poor adhesion and uptake problem on the surface and in the foliar applications, and also the penetration problem in the soil applications. So our technology also helps the extended adhesion and uptake of the plant tissues in the foliar application, and we can also protect our biomolecules for the soil-based applications. So in the playground of our technology, we of course started from the developing technology, but we choose crop protection area and also food preservation because of the actually the revolution of the biological molecules occurs in this industries. So in crop protection area, up to now, we have biofungicides, and also we are developing our bioinsecticides. And next year we plan to launch our bionematicide products. Our first product is the Micox. Micox is the generated from our baseline technology, and it's cost-effective, standardized efficacy, and residue-free, and it contains thymol, geraniol, and oregano oil, and it's directly have an effect on the specific plant pathogen fungi, and we approve in different plants. So when we compare with the activity in the field with the traditional botanical pesticide, so our technology and our products almost 30% higher than traditional applications, and we can we can catch the synthetic fungicide application period almost. So we tested our products with different partners via Crop Science and BSF, and also Hektaş from Turkey. So we have specific tests, field studies for the benchmarking test. Actually, we tested against with the regalia. It's the one of the important fungicide and biofungicide on the market, and we also understand the active standalone usage. So application interval almost 10 to 12 days, and the dosage rate of the regalia, the active dosage rate is 200 milliliter. So when we compare with the activity in the field, we catch the 70% disease prevention rate when we compare with the active dosage of the regalia. So when we increase the dosage of our product, we catch the 90% disease prevention. So we also want to understand the comparative benchmark test with the biopesticide, different biopesticide. The Serenade is the one of the important products in Turkey, which we use, generally used. It's the bacillus-based active ingredients, and almost they generally used 1.5 liters for 100 liter dosage. So we can catch the specific results, and almost same activity is 300 milliliter per 100 liter dosage, and it means the minimum 70% disease prevention rate. So our active dosage we offered to our customers is 500 milliliter per 100 liter dosage, and in this test, the disease rate was recorded 40% botrytis. So we also want to understand in the spraying program, the behavior of our product in the spraying program. So we compare with the Bayer crop science program, and we integrate our products after the flowering period, and we tested against a powder mildew. And we also, in one line, we as a single window, we want to use from flowering up to the harvest.
2 minutes. 2 minutes, Arda.
Sorry. So this in this test, we saw that Micox, then Micox used in the spraying program, the synthetic fungicide spraying program, after the flowering, catch the same efficacy of the fully synthetic program. So it means that we have very collaborative activity in the synthetic program. So it's very easy to use. The microcapsules I mentioned before, when they used to the products, crops, it's start to slow release and protect the fruits or the green body parts. So we have also post-harvest effect. Our products have an activity on the quality of the product. Also not for the vineyard application, for strawberry, and also for greenhouse applications. And with this technology also we produced a bio post-harvest product. And with this product we almost increased the shelf life of the nectarines and also peats almost 2 times higher. And we have, when we compare with the traditional usage, we have almost 50% higher activity when we compare with the traditional applications. And for grapes and citrus products. So our we are also preparing our bioinsecticide. It's called Talos, and it contains orange oil and neem oil, and also generated with our technology. And we have proven effect on the tuta absoluta in from larva 2 to larva 4, and we catch the higher kill rate in after 2 to 3 days in the greenhouse conditions. Also have an effect on the acars and the egg laying reduction on the acars to spotted spider mites actually. And we have a significant impact on the climate, and we can help to eliminate 65 tons of carbon dioxide emission when we compare with the chemical and synthetic fungicide-based applications. So we can we have value propositions. We have residue-free, multi-effective, stable, and user-friendly product portfolio. And also our discovery, we also have a workflow for the discovering of the biomolecules, and we are investigating biomolecules from the fungi, bacteria, and plants. And we have we found specific peptides, antifungal peptides from the fungi, specifically entomopathogenic fungi. And we have identified more than 100 peptides which directly kills the fungi and specifically for fungi, and no effect on the mammalian or cells or bacteria. And it's a very in the nanomolar and degree activity against specific pathogens, and it has its induced reactive oxygen production in the fungi and also regulated cell death. So we are preparing these products against specifically for oomycetes and also necrotrophic fungi. So we develop a recombinant-based expression system on yeast, because these peptides are cysteine-rich residue, have cysteine-rich residues and don't it can be produced by bacteria or specific algae. They very suitable for the yeast-based expression systems, and we hope to reduce targeting to reduce this peptides production from to actually up to 0.5 to €1 per gram with our manufacturing technology. Thank you for listening to me.
Thank you so much for that presentation. There's lots of information there. And just to remind everyone, we will be sharing all the presentations. So you will be able to look through Arda's presentation in full. Lots of interesting stuff. It's extremely interesting. I also like how you looked at the climate impact as well, which is really important. Other sort of plant pests and diseases that are really important in this region were mentioned. I heard tuta absoluta as well, which is something of interest to many in the room. Quite a few questions here for you, and we'll be quite quick, Arda, but here's a question around the waste. These I guess this new technology, is it biodegradable, or how does it end up in the environment?
Yeah, actually, our product, our microcapsules generated from the biopolymers. So this biopolymer is fully degradable in environment, and also it's safe for the consumption for the mammalians and also humans.
Yeah.
Yeah, it's generated from the blend of the fungi also.
Oh, excellent. When you apply it, does temperature or weather have an impact on the efficacy of the microcapsules? I mean, if it rains, or if it's too hot, or if it's too cold, I mean, does that have an impact on how the microcapsule breaks down or lets the product work?
So in hard conditions, of course, in the fully rainy conditions, if I understand the question true, but in the hardest conditions, the capsules also, of course, you have to spray, repeat your spray, of course, but it's the same as in all different biopesticides.
Yep.
But it's more, I can say it's more resistant to the specific conditions because the adhesivity is so higher than its adhesivity is so higher. I mean that to the plant green body parts.
Excellent. Okay, thanks. That's a good answer. And here's a question here from Cyril. Hello, I'm very interested on this product. Are they available here in the Asia markets yet? Specifically for Philippines, I would like to use it with entomic pathogens in the future. Where are you currently using this or trialing it? In which countries and regions, and what's your sort of interest in the future?
Actually, now we are currently in Turkey. In the next years, we want to extend our market actually to the Africa and also to the Asia. So, but now we are currently testing our products in the countries in nearby in the Mediterranean side as a testing. After the all testing results in different climate conditions, of course, we want to be in the market.
Yeah, excellent. And here's a question here: how effective could it be to protect maize grains against contamination by mycotoxins?
So we have a specific tests in with the company who is one of the biggest company in the world. We have a collaboration now, and we are testing our products in a different stage of the mycotoxin production. From we are going to start for the soil application against Fusarium for the maize, and then we are going to test our micro-encapsulation technology if suitable or not in the silo-based application. So we are want to understand from the field to the silo the full we hope the full protection system, and now we are testing our products in there.
Great. Well, it sounds like you've got a busy time ahead of you, Arda, and it's really interesting to see this, and it looks actually fantastic work. Looking forward to hearing more about it within the next year as well. We'll invite you back as well to see how progress is going, and hopefully I can see lots of people within this region interested to work with you as well, to see if they can maybe partner. So you've got a few questions there in the Q&A. So thank you so much for joining us.